Hi, I'm Judy Yen from the laboratory of Kate Rubins at the Whitehead Institute for Biomedical Research.
Here, we demonstrate how to label the antisense RNA samples collected from both Vaccinia virus-infected HeLa cells and virus by amino-allele coupling of dyes. We then show how to clean up the labeled aRNA and prepare it to be hybridized for microarray analysis of gene expression. Previous Joe videos highlight how to perform the viral infections, total RNA isolation, and RNA amplification and amplification steps.
So let's get started.
To begin aRNA labeling, first add 1 microgram of the aRNA samples into 1.5 milliliter microcentrifuge tubes. Vacuum dry the samples on low or no heat until they are completely dry. Cap each tube as soon as it is dry. It is important to not over-dry the samples. After they have dried, add 9 microliters coupling buffer to each tube and resuspend the aRNA by gently vortexing for 1 minute. Centrifuge briefly to collect the sample on the bottom of the tube, and then let the sample sit on ice. Next, add 22 microliters high-quality DMSO to each tube of Cy3 or Cy5 dye. 1 tube of dye is enough for 2 samples. The Cy3 dye is for labeling your reference samples, and the Cy5 dye is for labeling your test samples. Vortex the dyes to mix thoroughly. Remember to keep the dyes in the dark until ready to use. Do not prepare the dye earlier than 1 hour before using, and make sure no water gets in the dye DMSO mix at any point. Now, add 11 microliters of the prepared DMSO Cy dye mixture to each sample. Mix well by vortexing gently. Incubate for 30 to 45 minutes at room temperature. Cover the samples with tinfoil or keep them in a drawer to minimize exposure to light. After the incubation, add 4.5 microliters hydroxylamine to each sample to quench the reaction. Mix well by vortexing gently. Incubate for another 15 minutes at room temperature. Cover the samples with tinfoil or keep them in a drawer to minimize exposure to light. To clean the labeled aRNA, vortex the RNA binding beads briefly to obtain an even mixture before use. Prepare the aRNA binding mix at room temperature. Mix the binding mix well by vortexing. Aliquot the aRNA elution buffer into a 1.5 milliliter tube and incubate at 50 to 60 degrees Celsius for at least 10 minutes. Add 70 microliters of the aRNA binding mix to each sample and mix well by pipetting up and down 3 to 4 times. After mixing, transfer the samples from the PCR plate to a 96-well round bottom plate. Next, add 50 microliters of 100% isopropanol to each sample and mix well by pipetting up and down 3 to 4 times. Gently shake the plate on an orbital shaker for at least 2 minutes to thoroughly mix the samples. Move the plate to a magnetic stand to capture the magnetic beads. Leave the plate on the stand until the mixture becomes transparent and the binding beads have pelleted. Carefully aspirate the supernatant with a vacuum aspirator without disturbing the magnetic beads. Alternatively, carefully remove the supernatant with a pipette and discard the supernatant. The supernatant should be either a bright pink or a bright blue at this point due to the unincorporated dye molecules. Once the supernatant has been discarded, remove the plate from the magnetic stand. Now, add 100 microliters aRNA wash solution to each well and shake the plate for 1 minute on the orbital shaker at moderate speed. Beads may not fully disperse at this step. Return the plate to a magnetic stand to capture the magnetic beads. Carefully aspirate the supernatant with a vacuum aspirator without disturbing the magnetic beads. Alternatively, carefully remove the supernatant with a pipette and discard the supernatant. Remove the plate from the magnetic stand. Repeat the wash a second time with 100 microliters aRNA wash solution. After the second wash, dry the beads by shaking the plate for 1 minute on the orbital shaker at the maximum speed. Do not over-dry the samples. Elute the aRNA from the beads by adding 20 microliters preheated aRNA elution buffer to each sample. Vigorously shake the plate on the orbital shaker for 3 minutes, then check to make sure the magnetic beads are fully dispersed. If not, continue shaking. Once the magnetic beads have fully dispersed, move the plate to a magnetic stand to capture the magnetic beads. The supernatant contains the cleaned up, labeled ARNA samples and should be either a pale pink or a pale blue. Next, carefully transfer the eluted ARNA to a new PCR plate or PCR tubes. At this point, you can check the RNA concentration and the amount of dye in the samples by measuring 1.5 microliters on a nanodrop spectrophotometer using the microarray module. Immediately hybridize the labeled ARNA onto a microarray platform of your choice. Or alternatively, you can store the labeled ARNA at -80 degrees Celsius until you are ready for hybridization. This is a representative spectrophotometer reading and OD plot generated using the nanodrop spectrophotometer to measure sample concentration and dye incorporation. You can see the OD peaks at 260 nanometers for calculating the concentration of the samples, as well as the two peaks around 532 and 635 nanometers representing the Psi3 and Psi5 dyes, respectively.
We've just shown you how to fluorescently label amplified amino-allele incorporated RNA samples for microarray hybridization. When doing this procedure, it's important to remember to resuspend the dye in DMSO less than 1 hour before coupling and ensure no water gets into the dye DMSO mix, as it will react with the active group on the dye. It is also important to remember not to over-dry the RNA. If necessary, the samples can be dried down to 1 to 2 microliters rather than completely dry, and then resuspended well in the coupling buffer. During the coupling reaction, keep the reactions in the dark with occasional flicking and spin down if desired. So that's it. Thanks for watching and good luck with your experiments. Be sure to watch the other videos in this series, where we show how to perform a time course infection with the Vaccinia virus and RNA extraction of the samples, and the procedure for linear amplification of RNA samples.